Hi guys, on the third stop of our detailed tour of the cell, we are going to be discuss the last fourth functional categories of organelles, and this is the cytoskeleton, the structural support that are responsible for movement and communication. In early days, biologists thought that organelles of eukaryotic cells floated freely in the cytosol or the cytoplasm, but improvement in both light microscope and electron microscope have revealed the cytoskeleton. Cytoskeleton, this in yellow tubes, those are a network of fibers extending throughout the cytoplasm plays a major role in organizing the structures and activities of the cell. The most obvious function of the cytoskeleton is to give mechanical support to the cell and maintain its shape, provides anchorage for many organelles inside the cell, and even cytosolic enzymes molecules. Cytoskeleton also involved in cell motility movement, includes both cell location and movement of cell parts. And we have three main types of fibers that make up the cytoskeleton. We have microtubules, we have microfilament that built from actin filament, microtubules they built from globular protein tubulin, and we have intermediate filament. The first component of cytoskeleton, we will study or discuss the microtubules. This is the thickest of the three, and those are hollow tubes or rods, structures from globular protein called tubulin. Microtubules function is to shape and support the cell and also serve as tracks along which organelles equipped with motor proteins can move. Microtubules guide vesicles from endoplasmic reticulum to Golgi apparatus and from Golgi to plasma membrane. Microtubules also involved in the separation of chromosomes during cell division. So inside the cell, vesicles and other organelles often use motor protein. Those feed to walk. They walk to their destinations along a track provided by the cytoskeleton. Vesicles and other organelles too. These two from electron microscopy image. Here shows the vesicles. They use the microtubules as track to get their destination. Since microtubules or cytoskeleton involve the cell motility or movement, so we will talk about the cilia and flagella, these two structures responsible for motility with eukaryotic cells. Some eukaryotic cells have flagella and cilia. Those are cellular extensions that contain microtubules. Specialized arrangement of microtubules is responsible for the beating of these structures. Many unicellular processes are propelled through water by cilia or flagella that act as locomotor appendages. The sperm of animals, algae, and some plants have flagella. They can move fluid over the surface of the tissue. We have an example in our body. We have a ciliated lining of the trachea windpipe sweeps mucus containing trapped debris out of the lungs. In a woman's reproductive tract, the cilia lining oviduct help move an egg toward the uterus. Motile cilia usually occur in a large number. There's the green cilia cover the body of Paramecium. Uh, they occur in large number on the cell surface, and flagella are usually limited to just one or few per cell, and they are longer than cilia. Flagellum has an undulating motion like the tail of fish. In contrast, cilia have motion of cilia have alternating power and recovery strokes. So each motile cilium or flagellum has a group of microtubules sheathed in extension of a plasma membrane, the plasma membrane. 9 doublets of microtubules, those are 9 doublets of the microtubules, are arranged in a ring with 2 single microtubules in its center. This arrangement refers to as 9 plus 2 pattern. The microtubules assembly of cilium and or flagellum is anchored to the cell body or basal body. This is the basal body to the that are similar to the centriole. Basal body is responsible for forming or synthesize the flagellum microtubules or cilia microtubules. That's found in the base of each flagellum and cilia. And this is a adorable image from the scanning electron microscopy that reveals the cilia in our trachea cells. Since we are talking about microtubules, we should mention centrosomes. Centrosomes in animal cells, microtubules grow out from a centrosome. And centrosome, those are region that is often located near to the nucleus. You have an image for animal cells with centrosome. So centrosome is usually located near to the nucleus. And centrosome are responsible for forming microtubules during cell division. Anyway, within the centrosome is a pair of centrioles. This is centrosome. It's a condensed region near to the nucleus. And within the centrosome, we found 2 centrioles. Centrioles each composed of 9 sets of triplet microtubules. Each of them, each centrioles, is composed of 9 triplets of microtubules arranged in a ring. Centrosomes are associated with the nuclear membrane during cell division. Centrosome nucleated microtubules can interact with the chromosomes to build the mitotic spindles. We call those mitotic spindles during cell division, the spindles between the 2 centrosomes. They replicate themselves and then they divide apart to each pole of the cell, and then they will form microtubules or spindle microtubules that attach to each chromosomes. Centromere is copied only once per cell division or cell cycle, so each daughter cell inherits 1 centrosome. A centrosome is a cellular structure involved in the process of cell division. Before cell division, the centrosome duplicate, and then a division begins. The 2 centrosomes move to opposite ends of the cell. Proteins called microtubules, those assemble into a spindle between the two centrosomes and help separate the replicated chromosomes into the daughter cells. The second component of our cytoskeleton, we have microfilament. They are also called actin filament because they are built from molecules of actin, globular, those are globular protein. A microfilament is twisted, a double chain of actin subunits. The structural role of microfilament in the cytoskeleton is to bear tension, pulling forces. The three-dimensional network formed by microfilament just to the inside of the plasma membrane, we call it the cortical microfilament, helps support the cell shape. So microfilament are well known for their role in cell motility. Microfilament network gives the outer area of the cytoplasm called the cortex near to the cell membrane. This semi-solid consistency of gel in contrast with the more fluid state of the interior cytoplasm. As you see here in this amoeba diagram, it's revealed that the microfilaments in this area of the cytoplasm, we call it the cortex, is more semi-solid, or it gives the semi-solid consistency of a gel, and the inter area or part of cytoplasm is more fluid than the cortex. So microfilaments play a major role in the movement of cells. Here help the amoeba to form the pseudopod for movement or to form food vesicle. Microfilament also help with the movement of parts of cells, such as the contraction of muscles. Thousands of actin filaments and thicker filaments made of a protein made up from protein called myosin interact to cause contraction of muscle cell. Microfilament, the working of myosin projection, so-called the head of myosin, the purple one, the head of myosin. The head drives the parallel myosin and actin filament past each other so that the actin filaments approach to each other. In the middle, you see the red arrow. They will help to bring the actin filament to the middle of the cell. This shortens the muscle cell. Muscle contraction involves the shortening of many muscle cells at the same time. The third component, we have intermediate filaments. Intermediate are named for their diameter, which is larger than diameter of microfilament, but smaller than that of microtubules. Intermediate filaments are more permanent fixture of cells than are microfilaments and microtubules. Even after cells die, intermediate filament networks often persist. For example, the outer layer of our skin consists of dead skin cells full of keratin filaments. Intermediate filaments are especially sturdy and play an important role in reinforcing the shape of cell and fixing the position of certain organelles. For instance, nucleus sit within a cage of this cage is made of intermediate filament. The plasma membrane is usually regarded as the boundary of the living cells, but most cells synthesizes and secrete materials extracellularly to the outside of the cell. Although these materials and the structures they form are outside the cell, their study is important to the cell biology because they are involved in great many essential cellular functions. We have two types of extracellular component. We have cell wall for plants, and animal cells also produce extracellular matrix outside the cell membrane. In plants, we have cell wall outside the plasma membrane, and the cell wall is an extracellular structure of plant cells. This is one of the features that distinguishes plant cell from animal cell. The wall protects the plant cell, maintain its shape, and prevent excessive uptake of water. The chemical composition of wall, as you know before, and we studied with macromolecules or biological molecules, those built from cellulose, those are microfibers made up or made of the polysaccharide cellulose, embedded in a matrix of other polysaccharides and proteins. A young plant cell first secretes a relatively thin and flexible walls called the primary cell wall. We call it the primary cell wall. Between primary cell wall of adjacent cells is in the middle lamella. We have middle lamella between the primary cell wall for each cell or for adjacent cells. The middle lamella is a thin layer rich in sticky polysaccharides. We call it pectins. The middle lamella glues adjacent cells together. A pectin is used in cooking as a thickening agent in jams and jellies. When the cell matures and stops growing, it strengthens its wall. Some plant cells do this simply by secreting hardening substances into the primary wall. Other cells add a secondary cell wall because the plasma membrane and the primary wall. So the secondary cell wall would be added between the plasma membrane and the primary wall. The secondary wall deposits in several laminated layers, has a strong and durable matrix that afford the cell protection and support. Wood, for example, consists mainly of secondary wall. Although animal cells lack walls akin to those of plant cells, they do have an elaborate extracellular matrix. The main ingredients of the extracellular matrix are glycoproteins and other carbohydrates containing molecules secreted by the cells. Recall that glycoproteins are proteins with the covalently bonded carbohydrates. The most abundant glycoproteins in extracellular matrix uh, in most animal cells is glycogen. The collagen, the most abundant extracellular matrix or glycoproteins in extracellular matrix of most animal cells. Collagen fibers embedded in network woven out of proteoglycans secreted by cells. Some cells are attached to the extracellular matrix by extracellular matrix fibronectin. We call this glycoproteins fibronectins. And fibronectin and other extracellular matrix protein bind to cell surface receptor proteins called integrin. Those are protein integrin built within the plasma membrane or phospholipid bilayer that are built into this plasma membrane. So those protein integrated within the plasma membrane, we call them integrins. They attach to the fibronectins that hold the extracellular matrix. So on the other side, integrins or those glycoproteins are attached within the cytoplasm with the cytoplasmic microfilament. Okay, they attach with the microfilament in the cytoplasm. On the other side, they attach with the fibronectin that holds the collagen and in turn hold the most extracellular fluid. The extracellular matrix can regulate a cell's behavior. The extracellular matrix around the cell can influence the activity of genes in the nucleus. They affect cellular functions by mechanical and chemical signaling pathways. Mechanical signaling pathways involved with fibronectin integrins and microfilaments. Cells in animal and plant are organized into tissues and then organs and organ system. They form organ system. Neighboring cells often adhere, interact, and communicate via sites of direct physical contact. In plant, it might seem that the non-living cell wall of plants would isolate plant cells from one another. But in fact, many plant cell walls are perforated with the plasmodesmata, desmamine bond. Those are channels that connect cells or adjacent cells with each other. The plasma membranes of adjacent cells line the channels or the channels of each plasmodesmata and thus are continuous because the channels are filled with cytosol. The cells share the same internal chemical environment. By joining adjacent cells, plasmodesmata unify most of the plant into one living continuum. Water and nutrients can pass freely from one cell to cell. Cell junctions in animals, in general, in animals, there are three main types of cell junctions. They are tight junctions, desmosomes, and gum junctions. All three types of cell junctions are especially common in epithelial tissue, which lines the external and internal surface of the body. In our circulatory system and in our respiratory system, they are lined with epithelial cells, type of connective tissue. In tight junctions, neighboring cells or the plasma membrane of neighboring cells are very tightly pressed against each other, bound together by specific protein, forming continuous seals around the cells. Tight junctions establish a barrier that prevents leakage of extracellular fluid across a layer of epithelial cells. If you see here this type of tight junction, how seals or that form continuous seals around the cells, this tight junctions establish this barrier that prevent leakage of extracellular fluid across a layer of epithelial cells. This is an epithelial cells of our intestinal tract. The second type of cell junctions in animal cells, we have desmosomes. Desmosomes, one type of anchoring junction. A junction like rivets, fastening cells together into strong sheets. Intermediate filament made of sturdy keratin proteins anchor desmosomes and attach muscle cells to each other. In muscle, some muscle tears involve the rupture of desmosomes. The last type of cell junctions, we have gap junction, also called communicating junctions, provide cytoplasmic channels from one cell to an adjacent cell, and in this way are similar in their function to the plasmodesmata in plant cells. Gap junction consists of membrane protein extending from the membrane of the cell, of the two cells. These proteins create pores through which ions, sugars, amino acids, and other small molecules may pass. Gap junctions are necessary for communication between cells in many types of tissue, such as horse muscle and animal embryos.